Welcome to this deep dive into droplet microfluidics. Today we're going to explore how each tiny droplet becomes an isolated microreactor and why this enables ultra-high throughput screening, directed evolution, and combinatorial chemistry at unprecedented scales. Let's start with the fundamental question. What exactly is a microreactor in the context of droplet microfluidics? A microreactor is essentially a tiny, isolated compartment where chemical or biological reactions can occur completely independently from neighboring droplets. Think of it like this. Imagine you have 3 different chemical reactions you want to run. In traditional lab settings, you'd need 3 separate test tubes. In droplet microfluidics, each droplet acts as its own test tube, but at the picoliter to nanoliter scale. Typical droplet volumes range from 1 picoliter, that's 10 to the minus 12 liters, up to about 10 nanoliters, which is 10 to the minus 8 liters. How small is that really? Let's do a quick back-of-the-envelope calculation to put this in perspective. 1 picoliter equals 10 to the minus 12 liters, which is 1 times 10 to the minus 15 cubic meters. If we calculate the diameter of a spherical droplet with 1 picoliter volume, we get approximately 12.4 micrometers. That's about 1/10 the diameter of a human hair. Now here's where it gets impressive. If we divide 1 milliliter by 1 picoliter, we get 10 to the 9th, or 1 billion droplets that can fit in just 1 milliliter of total volume. Think about that. You could theoretically run 1 billion independent experiments in just 1 milliliter of space. This is the power of miniaturization. So how do we actually generate these droplets? The most common method is using a T-junction geometry in a microfluidic chip. In a T-junction, you have 2 immiscible fluids, typically an aqueous phase containing your reagents and an oil phase that acts as the carrier fluid. The aqueous phase flows vertically down, and the oil phase flows horizontally. When the aqueous stream enters the oil flow, shear forces pinch off tiny droplets at incredibly high frequencies, often in the kilohertz range. That means thousands of droplets per second. Now you might ask, why do we use oil as the continuous phase? Why not just make water droplets and air? The answer is that oil serves 2 critical functions. First, it prevents droplets from merging or coalescing with each other. Second, it prevents evaporation, which would change the concentration of reagents inside the droplet. We also add special surfactants to the oil phase. These surfactant molecules position themselves at the water-oil interface and provide a stabilizing barrier that keeps droplets separated even when they're flowing in close proximity. Let's talk about what makes each droplet a truly isolated microreactor. There are 3 key pillars of isolation. First is physical isolation. Each droplet is completely surrounded by an immiscible oil phase. Nothing can diffuse from one droplet to another because the oil acts as an impermeable barrier. Even if 2 droplets are flowing right next to each other, separated by just a thin film of oil, there is absolutely no mixing between them. This is physical isolation. Second is chemical isolation. Each droplet can contain a unique chemical composition. You can have droplet 1 with reagent A, droplet 2 with reagent B, and droplet 3 with a mixture of A and B. Third is temporal isolation. Each droplet experiences the same sequence of conditions as it flows through the microfluidic device, but at its own pace. You can control reaction timing with microsecond precision. Now let's connect this to ultra-high throughput screening. Why does droplet microfluidics enable such massive parallelization? Consider a traditional high-throughput screen. You might use a 96 well plate, which allows you to run 96 experiments simultaneously. Each well typically holds 50 to 200 microliters. Now compare this to droplet microfluidics. In the same time it takes to process one 96 well plate, you can generate and screen millions or even billions of droplets. Let's do another back-of-the-envelope calculation to really drive this home. A 96 well plate with 100 microliters per well uses 9.6 milliliters of total reagent volume for 96 experiments. Now 10 million droplets of 10 picoliters each uses only 0.1 milliliters total. That's 100 times less reagent for 100,000 times more experiments. The efficiency gain is approximately 10 million-fold. You're doing 10 million times more experiments per unit volume of reagent. This means reagent costs drop by orders of magnitude. For expensive enzymes, rare antibodies, or precious biological samples, this is absolutely transformative. Let's explore a specific application: directed evolution of enzymes. This is where droplet microfluidics really shines. The goal in directed evolution is to evolve an enzyme that has improved activity, better specificity, or enhanced stability compared to the wild-type enzyme. Here's how it works. You start with a library of maybe 1 million enzyme variants. Each variant is a slightly different mutation of the original enzyme. You encapsulate each variant into its own droplet, along with a substrate and any necessary cofactors. Each droplet becomes a miniature evolution chamber. The enzyme variant inside that droplet processes the substrate, and if it's a good variant, it will produce a detectable product. Typically, you use a fluorogenic substrate that becomes fluorescent only when the enzyme acts on it. The more active the enzyme, the brighter the droplet becomes. After incubation, you have droplets with varying levels of fluorescence. The dim droplets contain poor enzyme variants. The bright droplets contain the best variants that you want to isolate and study further. You can then use fluorescence-activated droplet sorting, or FADS, to sort these droplets at rates up to 10 kHz. That's 10,000 droplets sorted per second based on their fluorescence intensity. At 10,000 droplets per second, you can sort 36 million droplets in 1 hour. This allows you to screen an enormous library in a very short time. Now here's a critical question. How do you link genotype to phenotype? How do you know which gene corresponds to which enzyme activity? The answer is that compartmentalization ensures the linkage. Because each droplet contains only one gene variant, and the enzyme it encodes, the phenotype you measure, the fluorescence, is directly linked to that specific genotype. Whatever activity you measure in that droplet must have come from that specific DNA variant. There's no cross-contamination, no mixing, perfect linkage between genotype and phenotype. Another powerful application is single-cell analysis. Let's explore how droplet microfluidics revolutionizes single-cell studies. The challenge in biology is that cells in a population are incredibly heterogeneous. Even genetically identical cells can behave very differently due to stochastic gene expression. Traditional bulk analyses, where you lyse millions of cells together, averages out all this beautiful heterogeneity. You lose information about rare cell types and transient cellular states. Imagine a heterogeneous population, where cells have different metabolic states, different expression levels, different responses to stimuli. When you analyze them in bulk, you get an average that may not represent any individual cell. With droplet microfluidics, you encapsulate one cell per droplet. Now you can perform single-cell RNA sequencing, single-cell proteomics, or single-cell metabolomics on millions of individual cells in parallel. The encapsulation follows Poisson statistics. If you want mostly single cells with few empty droplets, you aim for an average of about 0.1 to 0.3 cells per droplet. The Poisson probability formula tells us the probability of getting K cells in a droplet, where lambda is the average number of cells per droplet. If lambda equals 0.1, about 9% of droplets contain exactly one cell, and 90% are empty. This is a good trade-off for many applications. Let's move to combinatorial chemistry. This is where you systematically explore chemical space by combining different building blocks. The goal is to discover new molecules with desired properties, whether that's a drug candidate, a new material, or a catalyst. The challenge is that chemical space is astronomically vast. Even with just 20 building blocks combined in pairs, you have 400 possible combinations. Add a third component, and you're at 8,000. 20 to the power of 2 equals 400. 20 to the power of 3 equals 8,000. 20 to the fourth is 160,000. The combinatorial explosion is real. Droplet microfluidics enables you to explore this vast chemical space by running thousands or millions of reactions in parallel. Each droplet contains a unique combination of reagents. You can inject different reagents into the microfluidic channels and merge them together in droplets in precisely controlled ratios. This gives you exquisite control over reaction stoichiometry. Let me give you a real example from drug discovery. Imagine you're screening for inhibitors of a particular enzyme that's a drug target. You have a library of 1 million small molecules. Testing each one in a traditional assay would take months and cost millions of dollars. In a traditional format with 100 microliter reactions, you'd need 100 liters of reagent. That's completely impractical for expensive enzymes or antibodies. With 10 picoliter droplets, you need only 10 milliliters total. That's a 10,000-fold reduction in reagent consumption. At 10,000 droplets per second generation rate, you can create all 1 million droplets in just 100 seconds. The entire screen can be completed in hours instead of months. Let's talk about temperature control and reaction kinetics in droplet microfluidics. One advantage of tiny droplets is extremely fast heat transfer. The thermal time constant scales with the square of the characteristic length. The time constant tau is proportional to L squared over alpha, where L is the characteristic length and alpha is the thermal diffusivity. For a 10 micrometer droplet, the thermal time constant is only about 10 microseconds. This means you can heat or cool droplets almost instantaneously. This enables rapid thermal cycling for polymerase chain reaction, or PCR, in droplets. You can perform PCR amplification in minutes instead of hours. How do reagents mix inside a droplet? This is governed by chaotic advection. At the microscale, the Reynolds number is very low, typically much less than 1. This means flow is laminar and there's no turbulent mixing. Reynolds number equals density times velocity times length divided by viscosity. At microscale, this is much less than 1. However, when droplets flow through curved or serpentine channels, they experience chaotic advection. The internal flow pattern becomes chaotic, leading to very efficient mixing. Inside the droplet, you get complex recirculating flow patterns that stretch and fold the fluid, leading to rapid mixing on timescales of milliseconds. An important practical consideration is droplet stability. Can you store droplets for extended periods? With proper surfactant chemistry, droplets can remain stable for days to weeks without coalescence or significant changes in composition. This enables time-course experiments where you generate droplets, incubate them for various times, and then analyze them. You're essentially freezing time for millions of reactions simultaneously. One challenge is Oswald ripening, where small droplets shrink and large droplets grow due to differences in Laplace pressure. But this can be minimized with appropriate surfactant selection. How do you actually read out the results from millions of droplets? There are several detection methods. Fluorescence is by far the most common detection method. You can measure fluorescence intensity, fluorescence lifetime, or even multiple colors simultaneously. Absorbance spectroscopy works for colorimetric assays, where the product changes color. Raman spectroscopy provides label-free chemical detection, allowing you to identify molecules without fluorescent labels. For the most detailed molecular identification, you can even couple droplet microfluidics with mass spectrometry, though this is technically challenging. Let's do one more detailed calculation about reagent cost savings, because this is often the most compelling argument for droplet microfluidics. Imagine you're screening potential enzyme inhibitors. Your enzyme costs $1,000 per milligram, and you need 1 micromolar concentration. Let's say the enzyme has a molecular weight of 50 kilodaltons. 1 micromolar in 100 microliters requires 0.5 nanograms of enzyme, which cost half a thousandth of a dollar per well. For 1 million tests in traditional format, that's $500 just for the enzyme, not counting other reagents, plates, or equipment. Now in a 10 picoliter droplet, you need only 0.0005 picograms, costing 5 times 10 to the minus $10. For 1 million tests in droplets, that's only 5 ten-thousandths of a dollar, or half a cent total for enzyme. That's a million-fold reduction in reagent costs. When you're working with precious samples or expensive reagents, this makes previously impossible experiments suddenly feasible. Now let's address an important question. What are the limitations of droplet microfluidics? No technology is perfect. First, channels can clog if you're working with large particles, cells that aggregate or precipitates. This requires careful filtration and optimization. Second, you need to ensure your surfactants don't interfere with your biological assays. Some surfactants can denature proteins or affect cell viability. Third, not all chemistries are compatible with microfluidic materials. Some organic solvents will swell or dissolve PDMS, the most common microfluidic material. Fourth, there's a learning curve. Setting up a droplet microfluidics system requires expertise in microfluidics, optics, and often some custom fabrication. Looking to the future, where is droplet microfluidics heading? The trend is toward fully integrated workflows. The ultimate goal is sample in, answer out. You load your sample, and the chip performs all steps: encapsulation, incubation, detection, sorting, and analysis. Imagine a single chip that takes your sample, generates droplets, incubates them with precise temperature control, detects a signal, and sorts the hits, all automatically. We're also seeing integration with artificial intelligence and machine learning. The massive datasets from droplet screens are perfect for training predictive models. Let me give you a real-world example of impact: Droplet Digital PCR for COVID-19 testing. Droplet Digital PCR, or ddPCR, partitions your sample into thousands of droplets. Each droplet either contains target DNA or doesn't. After PCR amplification, you count how many droplets are positive. Using Poisson statistics, you can calculate the absolute number of viral RNA molecules in your sample. The formula is viral copies per microliter equals negative natural log of the fraction of negative droplets times total droplet number divided by total volume. This method is more sensitive than standard quantitative PCR because you're counting individual molecules rather than relying on exponential amplification curves. Let me summarize why droplet microfluidics is truly revolutionary for high-throughput screening and combinatorial chemistry. First, the level of parallelization is unprecedented. You can run 1 billion experiments in the space and time it used to take for a few hundred. Second, reagent costs drop by factors of 1 million or more, making experiments with expensive materials suddenly affordable. Third, the perfect isolation of each droplet ensures robust genotype-phenotype linkage for directed evolution and single-cell studies. Fourth, the microscale enables fast heat transfer and mixing, allowing kinetic studies that are impossible in traditional formats. Fifth, digital counting of positive droplets provides absolute quantification without the need for standard curves. Here's the key conceptual takeaway. Each droplet is a complete universe unto its. It has its own chemistry, its own biology, its own fate. When you perform a droplet microfluidics experiment, you're creating millions or billions of parallel universes, each running its own independent experiment. This is the fundamental power of compartmentalization at the microscale. By shrinking the reaction volume while maintaining isolation, we unlock capabilities that are simply impossible at the microscale. As we wrap up, I want you to appreciate how droplet microfluidics exemplifies a broader principle in science. Miniaturization enables not just smaller versions of existing capabilities, but entirely new capabilities that emerge at small scales. The microscale is not just the macroscale made smaller. It's a regime where surface effects dominate, where laminar flow enables precise control, where heat transfer is nearly instantaneous. Droplet microfluidics harnesses these fundamental differences to create a technology that's transforming drug discovery, synthetic biology, diagnostics, and material science. Thank you for joining me on this deep dive into droplet microfluidics. I hope you now appreciate how each tiny droplet becomes a powerful, isolated microreactor, enabling experiments at scales that were unimaginable just a few decades ago.